Hey there, I suppose you are rocking with your day and you're studying well. You people are in class 10th now, and I hope you're learning everything in a better way. You know why? So, in this part, I have already told you this in the last part that what we are going to learn further. So, you know that already that we are going to learn the digestive system in human being, means nutrition in animals. So, I'm going to share my screen and let's start. Okay, so guys, into the life process, the first that we are covering is nutrition itself. So in this part, we will be covering nutrition in animals, but along with that, few part that was left earlier, we discussed about the factor that affect photosynthesis. Here we will be learning about how these factors are proven that, yeah, they are the one who are affecting the photosynthesis. Then in nutrition and animals, we will be learning about classification that I don't think we need to learn. We have done that. Nutrition in amoeba, nutrition in human being, and about the digestive system. So I'm trying my best to complete everything in this part. I'm already saying this, but in the next part also, the upcoming topic you will be learning there. But for now, let's concentrate on this. The first thing that we have to understand is condition to prove that sunlight, CO2, and chlorophyll, they are really very important in photosynthesis. Guys, the basic principle that is behind this activity is that you have to, or you may say you don't have to provide that thing. Like if you want to prove that sunlight is important, do not provide the sunlight, then you see the effect. Do not provide the CO2, then you see the effect. Do not provide the chlorophyll. I mean, not provide, but how come we just say that, yeah, chlorophyll is also the one that is helping in the photosynthesis? So there's a video with that. I'm going to show that thing. Okay. So in this video, they have explained by doing certain experiment. Let me just share my OneNote with you and show you that thing. And I don't think any external thing I have to do, just that I'll just explain thing through the video only. You'll be able to understand in a very beautiful way. So guys, what I'm going to do, I will play the video. Words are written below. I speak the same. You read the words. That will not be good now. So we have to just, be very calculative in this. So let me play the video first. Okay, I'll keep it on mute and I'll speak the words. That'll be fine. Huh? It might not be interrupting. So the first thing, see, they are saying conditions required for photosynthesis. Conditions required for photosynthesis topic we have covered in the last part. I don't have to show you these things. So directly let's come to the experiment part. So the first one is to prove that sunlight is very important. 1 second, let me just increase the size of this video. Let me just move back. I'll play this again. So the first thing that they are proving that sunlight is really very necessary. So in this video, what they are doing is they are taking a wide mouth bottle and they are, into the wide mouth bottle, they are covering it up. Like they say that they take, as they say that they are taking a bottle and then they are covering it with a black paper. Right. And then what it has written that they are covering the mouth of the bottle with the cork and they will be putting a cut into the cork. Now they are taking this plant and they are going to insert, not they are going to insert. You can also say that if you are doing the same, you can also do the same. Then what you have to do, just insert into the cut, into the cork, just insert the leaf still. And then let it be there. And this process should be done in the sunlight because in the sunlight photosynthesis is happening, and while you're putting this in the sunlight, when you suppose photosynthesis is happening, then you let it be and then you see what are the effects. After a while, when the day is over, you may say, or in the video they are saying after 2 days, even when the day is over, you can start getting the results. You take the leaf out and then you see. And they are putting it into the dilute iodine solution. Why they're putting into the iodine solution? Because when photosynthesis happens, starch is formed. And starch, iodine is an indicator of the starch, if you don't know. Clear indicator. That's why they're putting it into the iodine solution. And into the iodine solution, alcohol is there too, and they're heating it up so that it can be decolorized. So then, first of all, iodine solution, how they use, let me show you this part again. The person, or you can take alcohol and put leaf in it and just warm it up. After that, alcohol is an agent. I'm so sorry for this, guys. Alcohol is an agent that is going to remove all the chlorophyll. It is used to be known as the one which can just extract all the chlorophyll if you don't know. Then it will take all the chlorophyll out and the starch as well. So iodine is put. This is done for testing starch if starch is present or not. So starch will not be present. Half of the leaf which was, see, there's a point. I tell you one thing. In this video, they have not explained it up, but what you can do, the part that was there in the dark, cut it up. Half part you take. Half part which was outside there, starch might be present, and the one which is inside, starch will not be present. And all they have to be doing the experiment about they are doing is presence of starch. In first case, you add iodine. In second case also you add iodine after putting it in the alcohol. But in the first case, the transparent part or the decolorized part that you are using now, here starch result will be negative. And in this case, starch will be positive. Blue patch will be there. So that's what they are trying to do in the video. That's the first thing. The other vision not get any light, show no starch. Now the next experiment. The next experiment is chlorophyll. That chlorophyll, this is really very important to prove that chlorophyll is necessary for photosynthesis. Okay, so for this thing, what they are going to do, let's read it up. Take a vegetative, okay, take a vegetative leaf. A vegetative part is partly green. Actually, it's not vegetative. I'm so sorry. Vegetative is non-reproductive part of the plant. This is variegated, as in which is present in different colors. Variation is there. So a green part and a partly white part is there. Take that kind of leaf from a croton or any kind of plant wherever it can be available. So what they're going to do further? Now drop this leaf into the boiling water and let it boil for a few minutes for killing the cells in the leaf. Now boil the leaf in a rectified spirit over a water bath to decolorize. It's just that they are putting alcohol in it. So because alcohol, I have told you, just extract all the chlorophyll out of it. Then place the leaf in the dilute iodine solution. The green area of the leaf turn black color indicating the presence of starch. What he is saying that because chlorophyll is the one that helps in photosynthesis, they have taken the leaf wherein half of the leaf chlorophyll is there and half of the leaf chlorophyll is not there. So by this process, what they are trying to say that the part where chlorophyll is present, their starch is present because it is helping in photosynthesis. And the part where chlorophyll is not there, you do not find any result. That's the exact thing I would say. So it's almost the same experiment. What does this activity prove? I have told you that. You can just read this information out. Okay, so the next one is to prove that carbon dioxide, this is really necessary for photosynthesis. See, for photosynthesis, carbon dioxide is the one that is actually fixed, right? So insert a leaf in a potted plant in a bottle. Then pour some caustic potash solution into it and do not cover it with the black paper. Put it just like that. Caustic potash is used to absorb carbon dioxide from the bottle. Leave the plant in a place where it gets sunlight 2 days. Then plug the leaf and test it for the starch. The portion of the leaf which is inside the bottle will not turn black into the starch testing because it was deprived of starch. Here, starch did not form. Here, starch is not prepared because there's no carbon dioxide present in the bottle. The other portion of the, so point is that guys, they are taking the caustic potash solution and they're putting leaf in it because then it is going to actually absorb. It will take the CO2 and convert it into further hydrates. But point is that it will just absorb all the carbon dioxide. So inside the jar thing, even if it is outside, carbon dioxide is not available, which is taken up by the stomata. So if it is not available, then photosynthesis is not going to happen in that place, and that's it. So this is a clear-cut proof that CO2 is Also, really very important in photosynthesis. Fair enough? So we are done with the first part of this video. Now let's move forward and let's talk about nutrition in animals. I'm going to be again on the presentation thing, and here we have to talk further. So the next thing, guys, that we have to talk about is nutrition in animal. The basic information about the nutrition in animals I have provided you earlier, that nutrition is of two type, autotrophic and heterotrophic, and obviously in animal this is heterotrophic. And out of heterotrophic, this is mainly of a holozoic kind of nutrition, where food is taken as a whole inside the mouth and internally only digestion happen. Into our body, it will be digested with the help of all the juices, all the digestive enzymes. Holozoic nutrition. Fair enough? Now, further thing, if you remember, I told that this is of three type, herbivore, carnivore, and omnivore. I will not tell you the definition, and I'm telling it, I'm not telling. Okay, so the next thing that we have to understand, see, what is nutrition? What is the idea of nutrition? I don't think I have to give you that thing, because nutrition just absorbing nutrients that we use for forming energy for surviving. Now, the first thing in this part that we will be covering is nutrition in amoeba. So this is nutrition in amoeba we are going to discuss. Before I proceed with nutrition in amoeba, let me give you a little information about amoeba. You have learned diversity chapter in class 9th. Amoeba is actually the organism which is unicellular, and this is eukaryotic, and this belongs, they are aquatic. You will mainly find it in the water. But what I was about to say, that they used to be present in the second kingdom, which is Protista. Kingdom Protista. Into the kingdom Protista, there is a category which is known as protozoa. Into the protozoa, this one is present. Clear? So this is present into the protozoa of the Protista. Protista is actually the kingdom, and protozoa is a category of it. Now, talking about amoeba, this is like a unicellular organism which is not having a particular shape. Just externally it is covered, and whatever it eats upon these small bacteria or small food particle, it just ingests it inside. So the body is not having a particular shape. They show some cytoplasmic movement. The cytoplasmic movement is in them is known as cyclosis. Cyclosis, I repeat myself, cyclosis is defined as the cytoplasmic movement. So internally, a cytoplasmic movement happen. Cytoplasmic movement. Here, cytoplasmic movement happen, and they can change their shape and they can just take the food inside. I know that you have learned that in lower grades, but here let's understand what these things are known as. So internally, basically, if I tell you now the nutrition thing in amoeba, it used to happen by 4 steps. So the first one is ingestion. Second step is digestion. Third step is assimilation. And the fourth step is ingestion. Even before I explain you the diagram, ingestion is the one when food is taken inside. Digestion is when internal body is digesting it up with the help of enzymes and all. Assimilation is when all the nutrients are absorbed. Assimilation is when nutrients are absorbed. And ingestion is just throwing out the waste outside. Now, talking about the amoeba, ingestion is when, suppose this is amoeba with no particular shape and there is a food particle. So it will form a little, it will change its shape and take it inside. So there's a false arm that we call it as pseudopodia. Pseudo means false. Wherever you see the word pseudo, that means false. And podia means feet. So false feet are formed, which are kind of taking the food. So now they form food vacuole. Guys, into the 9th class also, when you were learning the cell chapter, you were learning vacuole as a cell part. So there are different type of vacuole. One of the vacuole is food vacuole also, that you can explain that this is a temporary vacuole. It's C, spelling is wrong. It's C-U-O-L-E. Acha, anyways, I'll just write it and then I will say whether spelling is right or not. What I'm doing, that's wrong. Second one is right. Okay. So basically, here it is taking the food inside. It forms food vacuole. Now, one thing that you have to remember, food vacuole, it is also known as phagosome. Food vacuole, it is also known as phagosome. Phagosome. Food vacuole is this one, and that is phagosome. Now, so this is ingestion, just ingestion. Now digestion. How digestion will happen? This phagosome, this is going to be the pink color thing that you see now. This is being digested. Why? Because here are lysosome. Imagine, not imagine. Acha, they have shown in this step. I didn't see the diagram actually clearly. Suppose there is a lysosome. You know, there are suicidal bags, lysosome, digestive bags present in the cell, which is full of enzyme. So into that lysosome will be present. This lysosome, it's having so many of the enzymes, it is going to actually add up. Suppose here lysosome is present. Now it is going to be mixed with it. Lysosome will mix with the phagosome and it will form phagolysosome. So I suppose I should call this phagosome. And because lysosome enzymes in this diagram, they have shown that with the food particle, lysosome enzymes are mixed. So to this, we call it as phagolysosome. Phagolysosome. So my point is that food vacuole is also known as phagosome, and once lysosome attaches with it, we call it as phagolysosome. So this is the second step, digestion. Third step is assimilation, where all the nutrients, it'll be like all digested internally. So here mainly third is absorption and assimilation. Actually, as absorption in the diagram they have written differently, but absorption you can write it as a part of assimilation itself, that all the nutrients are being absorbed. And then the last step is egestion, where the food, waste food, this is egested out. And that is all about the digestion in amoeba. And I suppose that's clear to you. You can just write a 5 marks question on it. Where you have to make the diagram. There are certain time question asked in the paper. Like suppose they ask nutrition in amoeba, explain. Guys, wherever you feel diagram is needed in bio, just make it. Don't wait for the question that they would be asking. If they're asking, then you have to make it well labeled. If they're not asking, just make it, but then make it less labeled, simple, easier to make, easier to understand. So that's nutrition in amoeba. Now the next thing that we have to talk about is nutrition in human being. Okay. So basically, as in amoeba also, it's all holozoic nutrition. Ingestion first, digestion internally, then absorption and assimilation, and then ingestion. Okay. Now, guys, one thing. We have a tube from the mouth to the anus. There is a single tube present, which is known as alimentary canal. Digestive system tubule thing is known as alimentary canal. So this alimentary canal is present throughout our body. And into this, while we take the food, there's certain movement happen, all through the alimentary canal. That is known as peristalsis. Peristalsis. So this 1 mark question can come in your paper, that what is peristalsis? So this is defined as the rhythmic contraction of the alimentary canal. Rhythmic contraction of alimentary canal. And then you can say that this helps in the food movement. Now, I have few videos over it too, but I suppose I'll explain you. Once we are done with the digestive system, I'll show you video. You'll be able to relate in a better way. Rhythmic contraction of alimentary. So throughout the mouth and anus, it's approximately 9 meter long tube. So I suppose that's clear to you. Now we have to see all the parts of the digestive system. Okay. So let's do one thing. I have a blank space written up there. So I'll come back there and I'll write then. But for now, let me show you this digestive system. I have tried to upload a picture that is having maximum labeling because we've learned that in 7th class. We are repeating it. So let's learn it in little detail. Okay. So first of all, there is the mouth part. So mouth further has different part. I'm going to come back to this diagram because I'm going to actually explain all the parts. So for now, just understand I'm telling you the labeling. Then first there is a mouth. After mouth, there is a pharynx. Pharynx is the place where your nose and your mouth, they're meeting up. There's a pharynx. Then pharynx, then there's a food pipe. Esophagus, that is the food pipe. Okay, so after food pipe, you see all the glands are present. I'm going to explain you this thing also. After food pipe, there is stomach. After stomach, after stomach ends, there starts the small intestine. So this part is a small intestine. It's having duodenum, jejunum, and ileum. This is all small intestine. Okay, I'll better write it up there. Not write up, I'm just lining it up. This is all small intestine, the one that I'm making black. It's all black. This is all small intestine. So small intestine further has three parts. I'll show you diagram of that too. Now, guys, remember one thing. The very initial part of the small intestine, we call it as the duodenum, which appears to be like this. It used to be having three things connected to it. One liver, second pancreas, and third gallbladder here. I have a diagram of that, separate diagram also. After that, there is a large intestine, this one, and large intestine, and then there is rectum, and then there is anus. These are all part of the digestive system. I'll come back to this diagram also because it's very nice diagram. Maximum labeling is there in the diagram. Acha. So now, guys, let's talk about all the part of the digestive system one by one. I have this video. Don't worry, I'll show you these things. So let's talk about all the part of the digestive system. First of all, let's talk about the mouth. So you may say mouth is the uppermost part. This is covered by two soft lips, and this is the one which gives passage for the food, the entry of the food. And the cavity which is present into the mouth, this is known as buccal cavity. The cavity present in the mouth, this is known as buccal cavity. Into the buccal cavity, now there are many things they are present. Let me see. I suppose I have a diagram. I'll explain you in the diagram. But into the buccal cavity, teeth, tongue, upper jaw, lower jaw, and then internally whatever is present. So in the diagram, you see there is this palate and palate, the hard palate. Actually, there's two palate. The front one is hard, and the back one is comparatively soft. While you touch it, it's very tingling. Uvula is the one when you open your mouth, and then there's a little thing hanging up there. That's uvula, and then there are tongue, and then there's the teeth, and yeah, into the mouth there are salivary glands also. I didn't add the picture of glands. Acha, I've added. So basically, into a mouth, a huge amount of saliva is formed in a day. If I talk about the buccal cavity, I suppose I'll talk about the buccal cavity here. I thought I'll talk about the teeth first, but that's fine. Buccal cavity, I'm writing it up there. This is also known as oral cavity. You may say above palate, and at the lower part there's a throat. Palate and throat is there, and then there is a hard palate, and then there is a soft palate. Into the buccal cavity, chewing of the food happen. Food is chewed here, and then into the buccal cavity, saliva is secreted, and this saliva, this is mixed. I'm so sorry. Yeah, it's just saliva in my mouth that created the problem, I guess. Okay, so saliva is there in the mouth, that is secreted. The saliva is full of a lot of things. It's having enzymes, it's having buffer solution, it's having microorganism killing lysozyme. Yeah, lysozyme is one of the enzyme present in it. While the time we eat the food now, the external food, many of the microorganisms are there. They are killed in the mouth itself. Lysozyme is the one. Into the saliva, the first digestion of the food starts in mouth. Carbohydrate starts even getting started to be digested in the mouth. There is an enzyme in the saliva which is known as salivary amylase. Actually, this is amylase enzyme. Depending upon where it is present, we call it as salivary amylase. Pancreatic amylase means in the pancreas. So amylase. Now, guys, wherever you see the word amylase, that means they are talking about the starch. So this is the one that acts upon the starch and it degrades the starch into the maltose. Please remember it. In a day, nearby 1 to 1.5 liter of saliva is produced. And saliva in a mouth that is produced by glands, which are known as salivary gland. And there are three pairs of salivary glands. Names are sublingual, below the tongue, submandibular at the mandibles here. Submandibular, now this is also known as submaxillary. Submandibular, I'm writing here. This is also known as submaxillary. Remember the name, guys, for NTSC especially. Then there is a parotid gland. Parotid is like on the back side here nearby. This is parotid. Sublingual below tongue, then this one, and then parotid is this area. You know, there is a disease named mumps. It's a viral infection. Mumps, Mumps. Mumps में क्या होता है? What happened? There is a swelling in the salivary gland. I had it in my master, mumps. My whole swelling was there. I used to cover my, with the scarf, and then I used to go to the college. It's very, Anyways, yeah, that's about the buccal cavity. One thing that is more in the buccal cavity is the teeth. So let's see this diagram. Let me show you this. This is nasal cavity. Do I have to explain nasal cavity there? No, not needed. Okay, in the respiration, I'll explain. So these are the lips. This is jaw, upper and lower jaw. And this is oral cavity. This is uvula. Pharynx, I'll explain you later. But yeah, that's pharynx, where the nose and this one is meeting. This is tongue. Yeah, okay. And these are the glands, parotid glands, submandibular and sublingual. These are the teeth. Now, guys, talking about the teeth part. Into the mouth, we know that we do have four type of teeth: canine, molars, premolar, and incisors. So these incisor are the one in the baby, they appear first. Incisor for cutting, canine are also for cutting. Then premolar are for tearing, molar are for chewing, and yeah, for holding the things up. Okay, talking about the canine in the animals, actually, into the carnivores, canine used to be well developed. Well developed. We do not have, because we are omnivore. Initially, if I talk about the evolution thing, we used to be carnivore. No, we used to be herbivore, and then we used to be carnivore. Yeah, when the evolution happened in Africa area, in Africa, it used to be very. I'm talking about evolution, but let me talk. So first of all, it happened in Africa. It used to be a hell lot of greenery there, hell lot of greenery. So our tropical forest. Then we were the one like ape-like animal. We were herbivore. We were on trees. We started to get on land. We were eating upon the food. We were eating upon the fruits, plant, leaves. And then gradually with evolution, the tropical rainforest forest started to get off. I mean, it started to get vanish. And then there were no trees in the same area I'm talking about. Then there were no trees. So humans started to be on land. And for food, they started to be eating upon the meat. And that's when the brain development also started. During this time, brain development was gradually also getting started up. And they were on the land, and that's the time when their canine also started to be very, strong. And then they were typically eating upon the flesh and meat. And then they got omnivore, frankly saying. So, but in another animal who are especially carnivore, they are having very strong canine. You must have seen on the Discovery or Net Geo channel. Okay, guys, so that's about the teeth thing. Now, let me tell you. There are adult teeth. In the baby, there are teeth. In adult, there are 32. In babies, there are 20. And then there is a middle stage where there's 24. There is a thing which is known as dental formula. Dental formula means, what is the dentition? Dental formula means the arrangement. Dental formula is like we write all the teeth types in a proper way, in a proper formula, in a proper form of a formula, so that we can just understand the dentition. Dental formula is a way to understand dentition, as in the arrangement of teeth. That will be a proper way to define. Okay, I'm going to tell you dental formula, but before that, let me tell you one thing, guys. We used to say sugar should not be taken too much, because then sugar can increase acidic amount in our teeth. So the covering of the teeth, this is known as enamel, which is the hardest part. This can easily be broken down with the acidity. With acid. This can be broken down by acid. Remember it. Let me change the pen color. Actually, what happened when we are taking sweets or something, there is a bacteria named Streptococcus mutans or Streptococcus varieties there that keeps on growing in a mouth. It eats upon to the food particle and then it secretes acid. That is the one that acts upon the teeth and starts degrading it. That process is known as tooth decay. I'm literally just touching my teeth right now. There's a deposition plaque. If there are calcium, phosphate, or some minerals deposited in the mouth, they form plaque. Here, just take it out. So that's why proper cleaning is done for the teeth. Teeth is really a very important part. You know, guys, what I have observed, whenever like mouth part is something, dental care is something that people do not take care until and unless there are major issues. Then they only go to the doctor. But taking care of this is really, really, really important. Let's talk about the dentition now. So, dentition is like writing the way, dental formula for human permanent dentition as in the arrangement. So canine, incisor, premolar and molar. Molar तो मतलब that is the wisdom tooth. बोलते हैं ना, we used to say that you got the wisdom tooth, so this like you are having the wisdom. So with age, sometime even in the age of 25, 30 age also, people do not have complete wisdom. So complete wisdom tooth. So this is the one that keeps on just getting out with the age. So anyway, now dental formula, this is incisor, canine, premolar, molar. How they have written in the bracket 2 means upper and lower, 2 times it is present. And here they have written as in two pairs. Two pair of incisor, two pair of canine, one pair of canine, two premolar and three molar. So then if you double it up, it will be like 2 into 2, 4, and then 1 into 2, 2, 2 into 2, 4, 3 into 2, 6, and then 4, 2, 4, 6. How much it is making? 4 to 8 for 12 and 2 times now I have multiplied it with the 2. Either you multiply it with the 2 as a whole or individually. It should be multiplied by 2. So that makes 32. Clear? I repeat myself, I have already multiplied it with the 2. Reason being, in the outside the bracket it is written upper and lower. So upper is all 16. 16, that makes total 32. So that's all about the dental formula. That's all about the types of teeth. Now let's move forward. Let's talk about the pharynx. Pharynx. This is like nearby 12 centimeter long funnel. And this is a common passage for food and water. Food and water? Food and air. That is the reason this is externally covered up by a flap. Externally covered up by a flap, and that flap is known as epiglottis. This place, epiglottis. While the time I'm drinking of eating the food, this is covering the windpipe. Or during the time when I'm not eating, so this is open and food pipe is closed. So that's why we say that while the time you eat, you should not speak too much. It might interrupt the food and air passage. You can just take it out, or the food can also take entry into the air pipe. That should not happen. So it just epiglottis. You may say if the function, it prevents choking of the food, not the food prevents choking of the food pipe or air pipe. Any kind of choking that happens. Okay. Now, talking about the pharynx, I have told you nose and mouth, they're meeting up at a common point. That common point is known as pharynx. And there are three parts of the pharynx. Nasopharynx is the nasal part. The nasal part. And oropharynx is the one nearby the mouth. And laryngopharynx is nearby the larynx. So three parts. And then it'll be meeting up further into the esophagus. You understand larynx. I'll explain you in breathing wala part. Larynx is the sound box thing. I'll explain you there. So for now, we are done with this pharynx. Let's move further. Let's talk about the esophagus. I don't have to tell you anything about the esophagus because here no digestion happens. Only food is moving with the help of the peristalsis. Guys, in certain books, you see the spelling of esophagus as O-E-S-O and in certain O-E-S-O, both spellings are correct. It is nearby 25 centimeter long tube and it is just meeting up to the stomach part. Just food is passing here. It is also known as food pipe. Now it is taking entry into the stomach, and let me tell you parts and structure of the stomach. I had a blank space. I have added blank space to write, but then I'm writing on this one screen, so it's fine. Chalo, take it. I'll just write talking about the stomach here. Guys, stomach. Stomach is the main part where digestion happens. We used to say we are suffering from acidity. There are reasons for it. I'll tell you. First of all, understand stomach is present. It is like a J-shaped organ. It is like a J-shaped organ, and into the stomach, highly muscular movement happen. Stomach is having a little elastic muscles. It's a muscular organ where enzymes and strong acid is present, which helps in the digestion. There are protein digesting enzyme, fat digesting things. Fat, though, is digested further into the duodenum. I'll explain that. But here carbohydrate digestion happen. Into the stomach, different food particle can stay for a different time. Mainly into the stomach, four things happen. One is storage of food. Second is partial digestion, because complete digestion obviously will happen in the intestine. I'll tell you why. Partial digestion. Next is mechanical churning that happen with the help of muscles, muscular activity. And the next one is regulation of chyme. Some people call it as chyme, but it's fine. Sorry, regulation is continuous of chyme. Let me change the pen color. Purple one. So, I have told you, stomach, it used to be having elastic muscles around it, J-shaped organ. Food is further broken down here. So if I talk about the storage, food can stay here for a different variable period of time. If I say carbohydrate into the stomach can just stay for 1 to 2 hours. And then talking further here, 1 to 2 hours, protein can stay up to 3 hours. I'll just exit to the presentation and then enter again, because you see my writing. Protein can be there for on an average 3 hours, and fat can be there for the maximum time period. It can ultimately, I mean, it can just stay up to 6 hours. Let me just exit the presentation and come back here again. Let me share my screen. Om Shanti. Yeah. So fat can stay for maximum time. It can stay for 6 hours into the stomach. Now partial digestion happen because into that glandular, we call it as the gastric juices. So there are gastric glands present. Wherever you see the word gastric, that means they're talking about the stomach. So gastric juices are secreted, that includes enzyme and acid that helps in the digestion. So they will be breaking down the proteins into the simple protein. They do not just see, proteins are made up of amino acid. It's a chain of amino acid. They do not convert the protein into amino acid, because complete digestion happen in intestine. They convert protein into the peptones. Peptone means simple protein. Protein as a high molecular weight is protein, but when I use the word peptone, peptide, that means less molecular weight wala protein. Protein with less molecular weight. Okay, so this is having lipase enzyme also. We call it as the gastric lipase, which eat upon to the lipid. Then there are carbohydrate digesting enzyme, protein digesting enzyme from on the walls. I mean, obviously there are glands present which are secreting it. HCl is also there. Now I tell you the function of HCl, though I'll explain that into the topic digestive gland. But there are many enzymes present in the digestive system which used to be present in inactive form. With the help of HCl, they are activated and then they start working. So HCl is doing two things. One, it is activating the enzyme. Second, it is killing upon to the any external pathogen or infectious agent if it is there in the food. Clear? That you can write in the function. Killing microorganism. Second, activating enzyme. sorry, activating enzymes. So these are the function. Now mechanical churning as in there happen periodic muscular movement into the muscles, which mixes the enzyme with the food. Simple. And then they form a semi-liquid form, semi-liquid form of the food. We call it as the chyme. Regulation of chyme means how it takes entry, how it is regulated. See, I am going to show you diagram of stomach and I'll explain these parts. I mean, not I'll explain, you will be understanding this because there's nothing I do not think you will not understand. Let's see the diagram of stomach now. Now, talking about the stomach, first of all, there are so many of the muscles present. This is muscularis externa, means external muscle layer. It is having three layers: longitudinal layer, circular, and oblique. See, it is having muscles and they are present in three design. One is longitudinal like this, then there is circular, and then there is oblique. They all are just increasing surface area while the time we eat. Now, internally, they can increase the surface area. So while they are normal, they appears to be like a folded skin internally. We call it as rugae. And muscle is internally muscle carrying. Sorry, I'm saying muscle. Stomach is internally protected by a layer which is known as mucosa. Mucosa as in mucus. Any thick layer that's present, like in nose, in the mouth, not in the mouth saliva, but internally. Yeah. So I was saying that into the muscles, into the stomach, strong acid HCl is present. It can harm the layers of the stomach. So that stomach is protected by mucus. Mucus is present throughout the alimentary canal. From mouth to anus, mucus is present. It does protective function. So here rugae of mucosa are present. They are secrete. Rugae means folding and mucosa as in it internally. हम बोलते हैं ना, we are suffering from acidity and there is a word which is known as ulcer. Ulcer means the mucus layer is getting degenerated and acid is directly coming in contact with the alimentary canal. That's ulcer condition. Anyways, and now talking about the part of the stomach, this is having fundus as in the initial part, and then cardia. Cardia is in part which is present nearby heart. Acha. So then there's a main body. And now talking further, there is a word that means sphincter. You must have heard the word sphincter. Wherever you listen the word sphincter in science, it means something which is like a wall, something which acts as a gate, opening, closing, something which is acting as in regulatory manner, opening, closing, regulating it. So the one which is present nearby the cardia, this one, we call it as the cardiac sphincter. So from the esophagus, when food is coming there, so cardiac sphincter, sometimes we vomit out because the cardiac sphincter open and this is just internal resistance of the food that just happened in regulation. Okay, so then there is a pyloric, the one that is present nearby the liver. So pyloric sphincter, we call it as pylorus. That is the one which is doing the fourth step that I mentioned, the regulation of the chyme. So this is the one that is regulating the chyme exit from here. Now it will be taking up entry into the duodenum. So pyloric, entrum, pyloric, so these are nearby the pyloric region. Pyloric as in stomach and the liver itself. So yeah, that's it. This is all about the stomach. Further, see, function of HCl, function of enzyme I have mentioned. Further about the further digestive glands, I'll explain you these things again, do not worry. For now, I'm just telling you all the detailed part of the digestive system. So the next part into the digestive system is, there are many blank spaces that I'm leaving. I will use this blank space for revising the stomach. Because I should write, because that's helping you to make notes. It releases gastric juice. Gastric juice include HCl, mucus, gastric enzyme. So gastric enzyme, it converts protein into the small protein, means peptones. Mucus, it does help in lubricating food. It lubricates food and also it protects the inner lining of the stomach. Talking about the HCl, I have told you two things. First, it activates enzymes. Second, it kills the microorganism. I don't have to write it. Let's continue. So that's a space I had to use. It's a revision for you also. Now the next thing is the small intestine. So from the stomach here, it's taking entry into the small intestine. Small intestine mainly include three part: duodenum, which appears to be like this one, like a C-shaped or U-shaped, whatever you say. Then jejunum and then ileum. Okay. Do I have more space? Okay. So now talking about the small intestine, this is like a highly coiled. Now talking about duodenum, this is the proximal part. I have told you this is like a U shape, and this is present nearby the pyloric end of the uh, stomach. Yeah. Okay. So this is also receiving secretion from liver and pancreas and gallbladder. How it is connected, I'll show you. Why I did not show you? I added the diagram. Huh? Guys, I added a diagram here. Just wait. I'm gonna show you how it is connected, the duodenum part. I'm gonna stop the screen share. How is it not possible? I'll just add the diagram. That's really very important. Pancreas, liver, and gallbladder. Because there's one thing that's really very important. It has come in the paper. It can come in the paper. Hmm. No, that's not well labeled. This one is well labeled. Common bile duct. This one is well labeled. Let me just add it up in the presentation. Huh? Yeah, so I'm going to explain you the small intestine, but before that, I wanted to add this thing. I have added the diagram. And what did I do? Blank dikh raha tha, I just added it. Let me just stop the screen share and just, edit the presentation. Okay. Control C, Control V. Didn't work? Yeah, cool. हाँ हो गया ठीक हो गया ठीक हो गया. Okay, so here is the written content for you. That also you must not lose. Actually, that's why it was a blank space. I added a picture. So the point is that here it will be like, suppose there was the stomach and it is taking the entry into the duodenum. Now about the duodenum itself, I am telling you why, because that's important. So right now I'm talking about small intestine only. That duodenum, this is connected with the liver. Liver used to have two lobes. Uh, Small and large, and yeah, into the liver bile acid is formed. Bile acid is formed in the liver, but it is stored into the gallbladder. Now into the gallbladder it is stored. This gallbladder is connected with the help of duodenum. Pancreas is also secreting enzymes for the digestion. Here also it is connected. The one thing that is important that can come in a paper is the name of the duct. See, this is right hepatic duct, this is left hepatic duct. That's coming from the liver. Now, from the gallbladder, the duct of the gallbladder is known as cystic duct. This cystic duct and all the hepatic duct, they all together meet to form the common bile duct. Then common bile duct meets up there, and this is pancreatic duct, and they are meeting up to the duodenum. कॉमन हेपेटिक डक्ट में लोग कंफ्यूज हो जाते हैं क्योंकि वो कॉमन हेपेटिक डक्ट को व्हाट दे इमेजिन कॉमन हेपेटिक डक्ट इज कनेक्टेड टू द गॉल ब्लैडर बट टू द गॉल ब्लैडर दिस इज सिस्टिक डक्ट टुगेदर दे आर फॉर्मिंग द कॉमन हेपेटिक डक्ट. See the diagram, see the labeling, really very important. So all secretion come up there and actual digestion, actual absorption also happen in the intestine. Now, moving further, I was talking about the jejunum part. Jejunum is also nearby 2.4 long structure. I should have added one more diagram into the jejunum. You have learned that in your lower grade also. Here, microvilli are also present. Into the ileum also that's present microvilli. Microvilli used to have ciliated epithelium as a tissue. Guys, I have not added diagram of the ciliated epithelium. Sorry, epithelium. I have not added diagram of the microvilli. I want to show it to you. So just hold on for a second. I'll show you diagram of the microvilli. So here, after this, I should add microvilli. Na? New slide. It's important. Actually, microvilli, because into the small intestine, into the mainly duodenum part here, food is digested, fat, carbohydrate, everything. But into the microvilli, absorption happens. Food is clearly absorbed into the intestine. Microvilli is forming an increasing surface area so that maximum, absorption can happen. Let me show you. So this is the diagram that I have added and did not share the screen. Too much into the lecture right now. Yeah, I suppose now you see the diagram. I've shared it right? Right. So this is like into the small intestine. Guys, like suppose a small intestine appears to be like a folded like this. But when you see it under the microscope, it appears to be like this diagram. There are villi present. Villi, when we see villi at a microscopic level, then they are microvilli. Microvilli, it appears to be showing a brush border. Brush border as in brush border. Simply increasing surface area. So blood vessels are in contact with the intestine. Externally, this is microvilli. That on the backside, blood vessel. Biggest point is that nutrients are absorbed into the intestine. So into the intestine, all the nutrients are absorbed and they go into the blood. They are filtered up there and go into the blood. So through the blood, everything then happen in our body. So here microvilli are present. They form a brush border, and on the back side here, lactase enzyme. I'll tell you that thing. So actually, now along with the blood vessels, there is also another lymphatic vessel. We call it as the lacteals. Lacteals actually it absorb fat. Like in the same way, blood vessel is there. There will also be a vessel which is known as lacteal. That is not a blood vessel, but lymphatic vessel. I don't know whether you know or not, but lymphatic system is a part of circulatory system in our body. In the same way blood vessels are there, in the same way lymphatic vessels are also there. You will learn that in the circulatory system of the same chapter. So there is a lacteal which absorb the fat, and that's why lactase enzymes are there. They absorb, take the fat, digest it, and absorb it up. But rest molecule just take entry into the blood vessels. That's all microvilli. And into the ileum also microvilli are present, but they appear to be like a club-shaped microvilli. That's all about the small intestine, guys. Now let's have a little discussion on the large intestine. Into the small intestine, all the food that is absorbed and food is digested also mainly into the duodenum. In the large intestine, water is retained. Water retention happen up there. And one more thing. Yeah. So the secretion of intestine, small intestine, is known as intestinal juice. Intestinal juice, this is also known as juice. This is also known as succus entericus. That is another name. Succus entericus. Clear? Now let's talk about large intestine. You're learning too much today. Okay. Talking about the large intestine here, into the large intestine, this is connected to the small intestine at the point of cecum. And here vermiform appendix is present. That's another point of discussion. But this is a colon. It used to have 4 or 5 parts. First is as ascending colon. Then there is a transverse colon, then descending colon and sigmoid colon, which appears to be like a sigmoid S shape. Then there is a rectum and then there is a anus. So large intestine, it appears to be like 1.5 meter long, LONGM long, Cecum is like the junction. The large intestine, it used to absorb water from the bolus, and then it store feces that we excrete. Until the time a feces that is excreted out of the body, this is being stored here. Now, guys, we should take fibery diet because fiber helps in making good feces. Feces can easily taken out of the body. That's why they are so cellulose, dietary fiber. They're mixed. So here, all the fibers are mixed with the excreta, so that it become hard and concentrated, and it can easily come out. And into the rectum, it is for a certain time into the rectum it is stored, and then it just take exit. Rectum muscles are also comparatively just able to increase surface area, but not too much. The exit of the exit of the food, it is controlled by anus sphincter. So there is also sphincter, anal sphincter. This is all about the large intestine, and with this we have discussed everything about the digestive system. Guys, digestive system used to have different glands. They are producing digestive juices, gastric gland, liver, stomachs, gastric juices, succus entericus. That's all I mentioned, salivary gland. What they are doing, what they are secreting, that's what I'm going to teach you in the next part also. But this is something that we have learned in detail, all the part. There's one more thing I'm going to explain you in the next part about the enzymes. All part, they all have different kind of enzymes and they're reacting in a different way. Guys, if a question come in your paper to write how we are digesting the food, all you have to mention are about the enzymes and explanation. Like in the mouth, it is having the saliva. Saliva is having, these are the composition of saliva. These are enzymes present. They are reacting in this way. And then you write about the further, it's taking entry. I'll explain this thing again once I show you those glands and list. So for that, I'll explain that in the next part. But for here, I suppose we are done and we are going to just have certain questions now. I suppose that everything is clear. What is the role of acid in a stomach? I don't have to say anything on the any question because I have told you everything. We have learned so much. HCl present in the stomach dissolve food particle and create an acidic medium. In acidic medium, protein digesting enzyme pepsinogen is converted into pepsin. I did not mention it, but now you know it. Pepsinogen is inactive form of the enzyme. Guys, I tell you one thing. If I'm using the word zymogen or proenzyme, they are always inactive enzymes. It used to be present in the stomach and they are activated with the help of the HCl. So pepsinogen is a type of zymogen, which is converted into pepsin, which further act upon the protein. HCl in the stomach act as a protective barrier against many disease-causing pathogens because it kills them. Next, how is small intestine designed to absorb digested food? I have explained that there are microvilli which increase the surface area. To make absorption effective, within the villi, there are numerous blood vessels that absorb digested food and carry it into the bloodstream. So you can mention that there are lacteals also. Lacteal, this is a type of lymphatic vessel that absorb fat. With the diagram, you can just make one more capillary kind of thing. So, we are done with the human digestive system. Further about it, we will be learning in the next part. So for now, I'm just stopping the screen share and I'm saying sayonara, take care, keep studying and keep smiling. Study is not bad. We don't have to take tension for anything.